Welcome to the Four Worlds podcast from Tomorrow's World Today. We're diving into the latest in tech, science, and sustainability, from nature's mysteries in the world of inspiration to the hands-on crafts of creation, the bold breakthroughs of innovation, and the scaled-up wonders of production. This is your ticket to the stories shaping tomorrow. And welcome back, everyone. Have you ever heard of biomimicry? So it's a design approach that views nature as a mentor, model, and measure for creating sustainable solutions. With us today is Benjamin Freedman and Phoebe Kwon from Limax Biosciences to discuss their Slug-inspired hydrogels. Before we get into all of that, thanks so much for joining us. How are you guys?
Doing well. How are you doing?
Great. This is another one we're excited for. We're really looking forward to this because it is an interesting concept. So what I will ask is if you just kind of introduce yourselves and tell me a little bit about Lemax before we really get into what you guys do.
Sure, sounds great. Well, thanks so much again for having us here. We're really excited to share more about our story with you. So I'm Ben Freedman. I'm a founder of Limax Biosciences. I'm a bioengineer by training, and it's a technology that I've been working on for a number of years now, which is really the culmination of much of the work that I completed during my postdoctoral fellowship in Dave Mooney's lab at the Wyss Institute at Harvard University.
And Phoebe, what's your background? How did you get involved in Limax Biosciences, and kind of a little bit about your story?
So I have a background in biology and material science, and I started working as a research tech at the Wyss Institute. And through that, I met Ben and started working on this project.
No, that's great. So you meet at the Wyss Institute, and then in 2021, that's where Limax Biosciences started hitting the ground running. So what was that inspiration behind getting this off the ground and that early vision from your time at the Wyss Institute?
So I came and started in the lab as a postdoc with a strong interest in tendon ligament biomechanics. And at the beginning, we were really focused on applying these materials to augment tendon and ligament healing. And those studies were progressing. And when everything started to shut down during the pandemic, we had essentially applied to one of these NSF core grants, which enabled us to start to explore many other applications of the technology beyond orthopedics. And through those customer discovery interviews, we started talking to experts in neurosurgery and dermatology and general surgery and plastic surgery. And with those calls, we started to realize the true platform potential of the technology. So when the pandemic started to wind down and we returned to the lab, we started undertaking a number of studies to really investigate the full platform potential of the technology. And through those discussions and new data that we started to collect, it became clear that there was some interesting commercial opportunity for the technology. We started getting involved with many different accelerators and incubators right on the Harvard campus, the Harvard Innovation Labs, and others in the Boston ecosystem. And through each of those engagements, we learned more about the translational potential of the technology, and it really motivated us to want to form a company in this space to try to take the technology and translate it to ultimately impact patient care.
And so when we're thinking about this hydrogel technology, and I say Slug-inspired, give me a little bit of background about that. When people hear Slug-inspired, where does that inspiration come from? How did you land on slugs as the foundation for this innovation?
Yeah, so we're coming from the Wyss Institute for Biologically Inspired Engineering. So we try to turn to nature for inspiration to try to engineer new materials and systems. So when we take a historical look back at existing bioadhesives and sealants, oftentimes researchers have focused primarily on generating strong adhesion underlying surfaces. You may have heard about muscle-inspired adhesives and other types of biopolymers. But these are confronted with one common limitation, that the materials themselves have poor cohesive properties. The matrix that comprises them is essentially weak and brittle, and they fracture. Even though they may adhere strongly, but the overall matrix, hydrogel that supports them, is weak and brittle. What we tried to do was turn to a different type of inspiration. We came across the slug, particularly the dusty Aryan slug, which is it's actually a really interesting, cool garden creature. Slug slime has high stretchability. You can take slug slime and stretch it about 10 to 15 times its length without breaking. It's a hydrogel system. It's about 90% water. It's composed of ions, proteins, and sugars, which give it these interesting tough properties. So we said, hey, this is much more of a stretchable, tough material. Maybe this would have different features if we tried to recapitulate them in our own materials. So without using any slow components, we tried to create materials that behave in a similar way. In the lab, what we did was we basically combined an existing hydrogel system that had been developed years before by some other former postdoctoral fellows in the laboratory. And we basically combined this dual network hydrogel with a liquid-based adhesive surface, which contains primary animates that work as float slime. And lo and behold, the system stuck pretty strongly to wet tissue surfaces. And we were surprised by how robust that adhesion was, and it really drew us down and led us down the path of many interesting studies and collaborations. And here we are today.
No, that's great. And with all of that background and coming up with that idea, taking the inspiration from slug slime, what was that maybe personal or clinical or even scientific moment that really sparked the need for this stretchable wet adhesive biomaterial?
Yeah, we tried to test these materials in all sorts of extreme conditions. And, one fun case for this is we had some active collaborations going on in some large animal studies. We stuck it to the surface of a beating heart of a pig. And that image and video still, I think, strikes everybody today when they see it for the first time. A material that's 90% water, that's highly stretchable, sticking to an actively beating organ. You don't see that with any other material system that's been developed to date. So, that I think has been a big aha moment for us as we look ahead towards what these materials can do and how they may potentially transform patient care.
Right. And Phoebe, with your background, what was that moment like for you when you, as Ben said, that aha moment? What was going through your mind when something finally clicked with this technology?
I've always been interested in improving patient lives, and just because the current standard of care is very lacking, and you could imagine like a band-aid being stuck on wet surfaces, like that doesn't really work well. So you can imagine like having like our hydrogel system stick in that environment and last and move with this dynamically moving tissue. That was just like sorry.
No, you're fine. Just a big moment for you guys, I'm sure.
Yes. It was a big moment for us.
So let's take it back. Limax Biosciences 2021, when things really started to get rolling. The fast forward to 2022, the Harvard President's Innovation Challenge. How did winning that award shape your belief and really, set the standard for this mission?
Yeah, it was we were there, we were on the stage. There were hundreds of technologies that were being initially pitched as part of the Innovation Challenge. And we were standing on the stage with four other amazing teams in the Boston Harvard ecosystem. Winning the challenge was huge for us to rocket and really kickstart our progress. At the time, we were just coming out of the laboratory, and having the support of the Burger Foundation enabled us to start to take on some of those challenges. We're incredibly grateful for that initial financial support as it really kicked off the initial program development for the technology on the startup side.
Right. No, that's great. That's, again, another huge feat for you guys. And, winning that, going into those early prototypes of the Lemax and this adhesive, how did those early prototypes kind of evolve from concept to, a physical adhesive that can have practical use?
There's a lot of iterations in material science. The initial formulations that we worked with years ago, we've been taking active steps to simplify. Do we need this component? Do we not need that component? How does this, viewed in the eyes of the FDA? We've made a number of iterations over the years. So, for example, the initial formulation wasn't resorbable. Now we've made it resorbable, and we've done other modifications to the system to further make it easier to handle for clinicians, whether it be what size or shape should be, how long should it take to get to set up, how should it be stored, what's the shelf life. There's all sorts of variables that come into play as we try to take it from an academic exercise into a viable product.
And I think that leads kind of seamlessly into my next question. And I'd love to hear both of your thoughts, because I'm sure there might be different answers from you, Ben, and from you, Phoebe. Some of those challenges in designing the hydrogel, that adhere so strongly but remains stretchable, and going through all of those variables. I'd love to hear some of those challenges that, both of you have faced throughout the development process.
Yeah. So where to begin? I mean, so let's talk about, making the materials degrade. There's different ways to make them degrade. Do you want them to go fast or do you want them to go slow? Independent of the bench science, what do the clinicians want? How should this vary for indications? All these areas are important points that you need feedback from end users. You need feedback from the FDA, and then of course there's scientific constraints. So I think, as we kicked off that arm of the project, all those variables came into play, continue to come into play, and because you want to make sure you're designing the product around the right form factor.
And what about you, Phoebe? Was there any maybe different challenges that you may have faced that might be a little different from Ben's, or did you kind of, feel those same hurdles as this process moved along?
I think it's similar. I think coming from an academic research to translating it out into actual bench-side use product is definitely takes a lot of iteration, and as Ben mentioned, needs a lot of user input. So that could completely change the design that as scientists and engineers we come up with. Yeah, it's like balancing the strength and the stretch and safe degradation. FDA is always looking to make sure that we're not putting toxic chemicals in our bodies, which luckily ours is bio-inspired, very bio-friendly.
Mm-hmm.
But other standard of care materials struggle with that a lot as well. So, being able to balance all of these different aspects of what goes into a medical device.
And when you just talking about all of those things that kind of go into it, I mean, let's say, guess what? It's not easy. It's not an easy process. So some of those challenges and getting over those hurdles is incredible. And when you're going through all of that, what were those design or functional, let's say, must-haves when you're translating this into a scalable medical product?
Yeah, so those are great questions. I mean, a lot of those depend on the end users.
Sure.
So, for example, if a must-have is that you must be able to cut it to any size or shape without it breaking or fracturing, right? Oftentimes, clinicians may want to do that on the operating field. That's a must-have. Another must-have, is it's not requiring any sort of cold chain or refrigeration. A lot of existing materials that have been commercialized require cold storage, and to overcome some of those limitations for transport, that might have applications both for civilian use as well as military DoD. Removing cold chain is a must-have. Shelf life. Some materials that have been commercialized, contain human dry products. They have, shorter shelf lives. We don't have that problem. So we can have extended shelf life and maintain performance. Another one, it could involve sterilization. There are some materials out there that, that can't be terminally sterilized, but ours can, and that enables new applications. So there's all sorts of must-haves when you design it. Of course, the materials have to perform well, and they have to, be superior to standard of care, and they have to be, in demand by clinicians. So we balance all those areas as we look towards new indications and applications. The list of must-haves or our target product profile is generated for those types of applications.
Yeah, absolutely. And, throughout this process, again, it's like a all hands on deck kind of situation, with a multidisciplinary team, from material science to clinical advisors. You two come from, different backgrounds and what you focus on. How did all of that shape the development journey?
Yeah, so I think it's all about the team. It's all about the team, and we look for folks to come on the team that share, of course, common interests and, collegiality, creating comfortable work environment and passion for what we're doing. But we also look for folks that can bring key skill sets and expertise. We like folks to feel validated as they bring those different areas to the team. So, we have a core of material science and bioengineering, I'd say, but we also seek clinicians that have a variety of different experience areas and different indications in dealing with different patient populations, and may have an eye for translation for how to design trials. We, go to top regulatory folks. We go to top folks that can support us in animal studies and reimbursement and quality management. So we're always looking for the best talent as we
as we built the team so far, and I look forward to building it even more in the future.
Yeah, absolutely. And that team, I'm sure, had a lot of hands in some of those, key scientific breakthroughs when we're talking about an adhesive that could be stronger than traditional sealants, that can stretch even more and work in wet and dynamic biological environments. So what were some of those key scientific breakthroughs as you explored all of that?
Oh man, there's been a number. I mean, they seem to happen every couple of years or so. Knock on wood, they'll keep continuing that way. I mean, the first one was, a stretchable hydrogel. We won't take credit for that. That was developed previously by other members of the laboratory. But just creating a hydrogel that was stretchable was previously not demonstrated. So traditional hydrogels are weak or brittle. You don't stretch them more than a couple tenths of length. So back in 2012, when the stretchable tough hydrogel was developed, well, here's a system you can develop that's developed. You can stretch it 20 times its length without breaking. That was a step function. The fact that you could then, it took another 5 years before we discovered that material could be sticky to tissues. And since then, we've seen an explosion in the field on developing tough hydrogel materials that can adhere. It's not just us; there's others that are working in the space, all built on this same foundational principle. Since then, we've had milestones where we've made these systems work as integrated drug delivery systems, where we've shown that they can not only attach to wet surfaces but be able to deliver payloads locally for extended periods of time. We've made milestones to make these materials resorb over away. We've created milestones to make them attach to each other in other areas instantly, as well as simplifying the formulation to remove the need for the addition of any other coupling agents added to the system during its attachment to tissues. So, not sure what the next big milestone will be, but these tend to happen with many and smaller breakthroughs along the way. So we're eager to see what we come up with next.
Oh, absolutely. I mean, there's a lot of potential here, and I can't even imagine some of the the breakthrough implications, the breakthrough potential that you hold in the future. And even just talking about that localized drug delivery, a big breakthrough there. What problems does that solve that maybe existing methods might not?
Yeah, we think there's a few areas for local drug delivery. I mean, one is the ability for it to be local. Mm-hmm. Unlike existing materials, they might just inject this material, can stick to the target tissue that you want it to go, and it will stay there for weeks. So we think that's one area that's useful. And we think that the fact that it can serve as a local depot for a drug or any drug or existing drug delivery system is another advantage. In terms of the amount of drug, we can load unprecedented amounts of drug into the gel. Most hydrogels are, typically pretty weak or brittle, as we talked about, and they're usually not loaded more than 1 or 10 mg per ml drug. We can load these materials up to 500 mg per ml drug. They still maintain their same stretchability and adhesion. So we're looking at order of magnitude improvement there, all using very simple drug release strategies. So we think all these areas may open the doors for some opportunity for these materials as advanced drug delivery systems.
Absolutely. And we talked about, some of the challenges getting into that and getting to that point, finding that localized drug delivery system. What about some regulatory and of course, maybe some scientific hurdles that you faced when bringing this new class absorbable adhesive to a class 2 or 3 device market? I'd love to hear about that, some things that people might not know when it comes to, any sort of medical development.
Sure. So for those tuning in that are new to the FDA space or just device regulations, so there's different ways that devices are classified. Of course, if it's a device versus a biologic, even within the devices, it's a class 1, class 2, class 3 device. The higher the class, the more scrutiny FDA will place on it. So our technology has applications in both class 2 and class 3 areas, as well as combination product devices. And depending on the class, it dictates how much testing is required for FDA approval, how much funding might be required to bring a technology to market. So we as part of our program development, we're pursuing both areas actively, and we've had several positive engagements with the FDA so far, and we hope to share more exciting news soon. So please stay tuned for some press releases in this space.
Yeah, absolutely. Again, we'll look forward to that, and I'm sure hopefully people will listen to this and keep an eye out as well. And so that is great. And just thinking about some of its capabilities and the wide range of different indications that it could do. How do you tailor Hemamax's tunable properties from those different applications, from minor wounds to internal hemorrhage control?
Yeah, it's a great question. So, I think that the performance is similar when we talk about the two. The difference is, how long that material may be in contact with the tissue for, and if it will need to degrade or be removed over time. So I think those are some of the biggest differences that we think about when we think about going from a class 2 product to a class 3 device.
Yeah, no, it is thinking about that. It is something that's spectacular, and how you even, how you get it to that point. It really is great. I'm really interested in the production process as well. Let's talking about scaling. How does that scaling production in the manufacturing process work? Is it in-house, leveraging partnerships? What does that kind of look like?
Yeah, so we are looking into various partnerships for this space. Of course, developing some things in-house, but also ways that we can further enhance our scalability while keeping the team lean. So we are taking some active steps on both fronts to be able to produce these materials at scale and for the variety of different demands that will be necessary, or the volumes that are going to be necessary to meet the clinical demands.
No, yeah, that's great. And we talked about the future a little bit. I'd love to know, what does, let's say, it's so cliche, but that 5-year timeline look? Where do you see yourselves in 5 years? What's the future? What are the next goals that you guys are trying to hit?
I think the next goals we're trying to hit are to finalize preclinical trials in key surgical models, and then move into early feasibility human clinical studies, and run pilot programs with surgical teams to refine usability.
And ultimately, we're trying to get these materials into the hands of clinicians. So we're going to be looking towards, at least one FDA approval in the coming years, and hopefully some steps where we can then start to scale the technology and get it into impacting patient lives.
Yeah, absolutely. Is there, as we wrap this up, this was great for one thing. I mean, something like this and its capabilities is something that should make, a lot of people excited. Just, what you guys are doing, what you've been able to accomplish thus far. It really is great. What else would you guys want to add that maybe that we left off that people can kind of look forward to, kind of get excited about?
Yeah, I mean, if you're listening in, you're interested in joining our mission, please do reach out. We're always looking to share what we're working with others, and we're always looking to grow the team and, of course, fundraise for our next endeavor. So if there are anybody that's tuning in that's interested in learning more, please don't hesitate to reach out.
That leads me into my next question. How can people stay in touch, keep up to date on all of the latest news from you guys from Limax Biosciences? Give the people a little bit of information of how they can maybe reach out or continue to follow along with the journey.
You can find us on limaxbiosciences.com, or also follow us on LinkedIn, and you can subscribe to our website and reach out.
Yeah, no, that's great. We're looking forward to ourselves following along with the journey. We'd love to work together again. And I just can't thank you guys enough for taking the time out of your day and joining us on the show.
Well, thanks again for having us. Look forward to keeping you posted with all of our progress in the months to years ahead.
Yeah, absolutely. Definitely keep in touch. We're excited. We're excited about the future for Lemax.
Well, thanks very much again.
Yeah, absolutely. The pleasure was all ours. All right, everyone, that is all the time we have. We'll see you next time. Thanks for listening to this episode of the Four Worlds podcast. Until next time, you can catch up on the latest innovations shaping our world at tomorrowsworldtoday.com. Follow us on Facebook and Instagram, and be sure to subscribe to our YouTube channel.